Now we focus on chemical buffers. The buffers usually function almost instantaneously, and it acts through a combination of a weak acid and/or a weak base and its salts. It is the first defense mechanism towards imbalance, and its actions aims to resist changes in pH. And here we have three main buffer systems: the bicarbonate system, the phosphate system, and the protein system. The bicarbonate buffer is usually formed by sodium bicarbonate and carbonic acid. The body tends to maintain a 20:1 ratio of bicarbonate to carbonic acid, and this can be elaborated in the following two equations. The hydrochloride acid will bind with sodium bicarbonate to form carbonic acid and sodium chloride. The carbonic acid also can bind with sodium hydroxide to form sodium bicarbonate and water. What about the phosphate buffer? The phosphate is a major intracellular buffer. It has a major role in the elimination of hydrogen via the kidneys, and it is essential within the red blood cells. It assists in the exchange of sodium for hydrogen and has the following reaction to compensate the excess hydrogen ions. Usually the hydrogen will bind to hydrogen phosphate to form a hydrogen phosphate ions. This can combine also with hydroxyl ions to form water and hydrogen phosphate ions. What about protein buffers? They include amino acids and also the hemoglobin buffer system. Hemoglobin usually works in the blood and interstitial fluid, and usually it will bind the carbon dioxide, binds and transport hydrogen and oxygen, and participate in the chloride shift on the cell membrane, and maintains a blood pH as hemoglobin changes from oxyhemoglobin to deoxyhemoglobin. What about amino acids? The amino group usually accepts a hydrogen ion. The carboxyl groups gives usually a hydrogen ion, and the side chain that can buffer hydrogen are usually present in over than 27 known amino acids. In this illustration, we can see within the red blood cells, the reaction usually, the carbonic acid can dissolve into hydrogen ions and bicarbonate. And if the hydrogen ion dissolve and pass through the red blood stream, usually it goes to the kidneys and will stimulate the kidney to increase the rate of hydrogen secretion in order to correct the pH. On the other hand, this also can stimulate the respiratory system in order to adjust the rate of breathing. If the carbonic acid through carbonic anhydrase is dissolved into carbon dioxide and water, this can also pass to the bloodstream, and it circulates and goes to the respiratory center through the brainstem to adjust the respiratory rate and probably increase the carbon dioxide given away. Now we come to a very important equation, which is the Henderson-Hasslebalch equation. And this is usually an equation used between the pH and the bicarbonate carbonic acid buffer system in the plasma, and it helps to calculate the pH. The equation states that the pH is equal to pKa plus the log of a molar concentration of a conjugate base divided by the molar concentration of undissociated weak acid. pKa is defined as a negative log of the ionization constant of an acid, while the pH is the negative log of the hydrogen ion concentration.